Today we will discuss about how this DiaBZl will cause increase in the TCR signaling. TCR means the T cell receptors that are engineered in the T cells. And the T cell receptors that we are discussing about is Ig4 HAT. It is Ig4 high affinity receptors that can detect tumor cells more efficiently and can trigger the immune response against those tumor cells. So in this video we will discuss how this DiaBZl will cause or will result in the increase in this TCR signaling. In the previous video we have understood that DiaBZl will cause this or DiaBZl will trigger STING signaling pathways in the T cells. In this video we will discuss how it will enhance the T cell signaling. So in order to check this DiaBZl effects on T cell signaling, they check uh, some of the phosphorylated proteins that results in the increase in the T cell response. That is phosphorylated ZEP70 and P38. Both of them shown to be increase in the cells which are treated with this DiaBZl. This indicates that this DiaBZl will has some role towards enhancement in this T cell receptor signalings, and the quantifications of this phosphorylated ZEP70 and the phosphorylated P38 is shown in this panel B, where we can see that after 3 hours we can see the increase in this phosphorylated ZEP and phosphorylated P38, and both of these indicate that there is an increase in the T-cell signaling in the case where cells are treated with DiaBZl. And thereafter they will check one of the pro-inflammatory cytokines such as IFN gamma, and what they have find out is that after 3 hours the difference between control and the DiaBZl is not statistically significant. However, after 20 hours they can see that difference is statistically significant. So this indicates that in the long term the DiaBZl will cause increase in the pro-inflammatory cytokines such as IFN gamma, and thereafter you need to check whether this DiaBZl has some negative effect on the T cells or not. So before moving into this panel D, one thing I need to say about panel C is that the panel C results are being done when T cell receptors are co-cultured with the tumor cells. Then only there is a increase in the IFN gamma will show, as the T cell receptors will recognize the antigen on the tumor cells, and then it will trigger this release of this IFN gamma. And after 20 hours we can see that the increase in the IFN gamma in the DiaBZl case is higher compared to the control case. So this indicates that DiaBZl has a role towards increasing T cell signaling against the tumor cells. So now we will move to the panel D, where they have checked the cytotoxicity effect on the DiaBZl, and what they have find out is that after 3 hours they are not able to see any statistically significant changes between the control case and the DiaBZl case on the cytotoxicity, and they have measured cytotoxicity based on the LDH, this lactose dehydrogenase, which are usually found inside the cells. And if there is a disruption in the cell health, then this LDH will move into the cellular matrix and will be detected with the help of the fluorescence. So in the case where DiaBZls are treated, the T cells treated with the DiaBZl for 3 hours, they do not see any changes or statistically significant changes in the cytotoxicity between the DiaBZl and the control case. However, after 24 hours, what they have find out is that there is a statistical changes that they have observed in the case where cells are treated with DiaBZl for 24 hours compared to the case where cells are not treated with DiaBZl. So indicates that and after that they also check the anti-apoptotic factors in the T cells at different time intervals. What they find out is that at 3 hours there is an increase in this anti-apoptotic factor, that is BCL2, but they see that at 20 hours there is a decrease in the anti-apoptotic factors. So this could be the reasons why there is an increase in the cytotoxicity after 24 hours. So combining all these results, it indicates that for a short term DiaBZl treatment, it will have less cytotoxicity effect. At the same time, it will cause increase in the pro-apoptotic factors that can enhance this TCR signaling against the tumor cells. I hope you understood how they have checked this DiaBZl's relations with the T cell signaling through these results that are presented in this figure. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.